Hi class. Now we are going into the last chapter of this course, Bioforce 61. And the title of the chapter is Carcinogenesis, Regulation of Cell Cycle, Oncogenes, and Tumor Suppressor Genes. And in the book, this is as chapter 18th, from page 603 to 640. And on our syllabus, this is mentioned as chapter 9, right? So let's go into this chapter. So essentially, if you note, every living organism, it wants to divide, right? And it is also not surprising that bacteriophages that infects exclusively bacteria, they infect, they also, the goal of infecting bacteria is to reproduce. If, let's say, if one bacteriophage is infecting one bacteria, the goal is to come out of the bacteria using the bacterial resources into million copies. So even bacteriophages and viruses, they are in between living and non-living, and they need a living cell to divide. The goal is to create, tons of progeny and to increase the infection rate and survival. And if we go to bacteria, bacteria also has the urge to divide all the time, but bacteria is limited by nutrition. So the basic point that I want to mention here is that every living organism has the desire to grow and reproduce and develop and differentiate in case of multicellular organism, such as human being, right? The whole story of human development is cell division. And even when a zygote develops into a mature adult, a lot of cell division is needed to maintain the body. And there are some cells in our body, for example, red blood cells, they have limited half-life. They come, they do their work, and they are removed from the system. The new cells come in. Our hair follicles, intestinal lining, all these cells are replaced even after we have developed into an adult. We need continuous cell division. But remember one thing, that on one hand, there are cells like skin cells or hair follicle or intestinal lining that keep growing and supporting our body. There are some cells in the organs, such as brain or heart, that they divide very slowly. So there are different profile of cells depending upon their ability to divide. Nevertheless, the cell division is the essence of our life, because our life journey starts with single cell, and as days, weeks goes by, we develop into an organism, a full-fledged human being. In this slide, it's a very interesting slide, which suggests that, which shows that a single cell can divide into two, and then these two cells can divide into 4 cells, and so on, so forth. So division could be arithmetic at a certain stage, then it becomes a logarithmic, depending upon the number of cells. And in this figure, try to appreciate that there are 3 different cell types, as shown by the colors of the graph or the line. And what you can see here, that if there are 3 different cell types, they have different growth properties. For example, cell type 2, which is green, they divide very slowly, right? So how do you say different slowly? So this is the time on this x-axis, and on the y-axis is percent confluence, right? So this is only 20% confluent on 144 days, after 144 days, on 144 hours, right? And if you see red cell, which is cell type 3, it remains slow until 48 hours, and then the division becomes from 10 to 20, and then by 96 hours it becomes 40. So the doubling time increases by time. So look at the difference between the green line and red line. So the point I'm making is that different cell types have different growth requirement to sustain itself. Generally, the cell division or cell growth can be measured like this. It has a lag phase uh, before the cell adjusts to the environment, and then it starts doubling and goes into logarithmic phase, and then it plateaus down, and then depending on the cells, it can divide, it can die, or it can stay as it is. So which cells can die? For example, RBC cells, right? So bone marrow cells, they develop and divide, and they reach a stationary phase, do their work, and then they're removed from the system. So the story is that each cell, they have desire to divide, they grow at their own pace, right? So what is that mechanism that supports cell division? And that is referred as cell cycle. Right? So cell cycle can be divided into two phases, M phase and interphase. M phase refers to mitotic phase or mitosis, which can be divided into two parts: karyokinesis, which is nuclear division, and cytokinesis, which is cytoplasmic division, and the product is two cells. On the other hand, interphase is made up of three phases: G1 phase, growth phase 1; S phase, which is synthetic phase or synthesis phase, where DNA replicates, chromosome duplicates; and G2 phase. G2 phase is before, again, growth phase 2, and G2 phase is before mitosis. The most important point here is that there are some cells which could be in cell cycle for, let's say, skin cells, but there are some cells which divide either very slowly or they have to perform very specialized function. For example, nerve cells, right? So they're not dividing all the time like skin cells or bone marrow cells, but they enter into differentiation phase and they lose their capacity to divide. So they are called in resting phase. Now here I would like to caution you, resting phase could be of two types. One, that cell loses its desire to divide, and it has entered into full specialization phase, such as nerve cells or heart cells, or cardiac cells. And there are some cells in the resting phase also, which are there and lying latent, doing its function. But if there is any trigger to these cells, then these cells need to divide. And for division, then again, the cells need to enter the cell cycle. And they enter through G1 phase, S phase, G2 phase, and mitotic phase. So let's look at each phase. So G1 phase, cellular content excluding the chromosomes are duplicated, right? In the G1 phase. That's the main feature of the G1 phase. In the S phase, the DNA, in the S phase, the new DNA is synthesized, DNA replication occurs, and also the chromosomes duplicate. In the G2 phase, because G2 phase is the phase before mitosis, during this phase, cells organize proteins, tubulins, microtubule, the organelles which have to be duplicated. They're all lined up for the mitotic, for the cells to enter into mitotic division. And mitotic division is typical mitosis, which is comprised of prophase, prometaphase, metaphase, anaphase, telophase. So these are karyokinase. Here, the nuclear division takes place, chromosomal division takes place, which is followed by cytokinesis, where cytoplasmic division takes place, and two independent daughter cells are formed. So once again, I would like to emphasize that there are some cells which may not divide. So these cells are highly differentiated cells like neuronal cells or heart cardiac cells, and they enter into G0 phase. But there could be regular dividing cells, for example, intestinal linings, skin cells or hair follicle, and bone marrow cells. Now, the most critical aspect is that how does a cell cycle follows? Does it follow blindly, or is there any rule? So the rule is that during transition, like from G1 to S phase and then S to G2 phase, there are some checkpoints, right? These are checkpoints. These checkpoints, they help to look for cells if there are any abnormalities. If there are any abnormality, then these cells are taken out. They have a potential to take out the cells so that there is no diseases. So G1 checkpoint, it checks the cells for cell size, nutrients, growth factor, and if there are any DNA damage. In G2 checkpoint, they look for cell size and whether the DNA has been replicated, right? If there are any problem, then cells can exit the cell cycle. But if the cells reach certain stage, cells cannot go back. And then during mitotic checkpoint or spindle assembly checkpoint, the cells are checked for chromosomal attachment to the spindle. Because if the chromosome don't attach to the spindle, then cells will, the chromosomes will not separate during anaphase, right? So these cell cycles, this cell cycle, advise has two phases, mitotic phase and interphase. Interphases G1, S, and G2, depending upon what kind of support a cell needs for cell division. A cell cycle, a cell can be normal, so it can go through this G1, S, G2, and M phase. Otherwise, these cells have to be checked at every phase that there are no problem. Because if there are problem in the cells and cell cycles let it go, the cells will be the starting point for the formation of cancer or tumor cells. So there are checkpoints, G1 checkpoints, G2 checkpoint, and M checkpoints, right? And each checkpoint, it checks the cell for various criteria, right? So let's look at again. Cell growth checkpoint, it occurs towards the end of phase growth, phase 1. Checks the cell is big enough and has proper protein synthesis phase. If not, then the cell goes through the resting phase until it is ready to divide, G0 phase. Then DNA synthesis checkpoint. It occurs during synthesis phase and it looks for the chromosomal division. And then mitotic phase to look for spindle fiber attachment and so on and so forth. So G0 could be a phase where the cells are differentiating, cells are getting repaired if there is any abnormality, or cells are doing its function until there is an abnormality. So G0 phase is very, very important phase for the for the health of the cell. Now, after this cell cycle, I would like to introduce to you some terminologies. Number one is proto-oncogenes. Proto-oncogenes are those genes that support the growth of the cells. Right? Proto-oncogenes support the growth of the cells. They are a stimulus. They are accelerators of cell growth. If these proto-oncogenes, if there's a mutation, then they become oncogene, and the growth of the cell becomes abnormal, uncontrolled, and that's the beginning of the formation of a tumor. So the oncogene is the mutant product of proto-oncogene. Now, just like I mentioned to you that proto-oncogenes are accelerated, tumor suppressors are those proteins which put a brake. They try to repress the cell cycle. They try to block the cell cycle such that there can be the abnormalities in the cell can be repaired, right? So proto-oncogene and tumor suppressor, they have opposite functions. Their job is to correct, to induce the growth or suppress the growth. Induce the growth is the necessity of life, but the dangerous part is that it can be mutated to, the proto-oncogene could be mutated to an oncogene. And and tumor suppressor also, which has the ability to suppress the growth to correct the error. If there is a mutation in tumor suppressor, then the defect in the cells will be carried down. So mutations in both proto-oncogene is a problematic, in tumor suppressor it is also problematic, because there will be no checks and balance mechanism if the tumor suppressor is mutated. And then the checkpoints. Checkpoints are those phases in the cell cycle at the boundary of the transition phase, for example, G1S or S to G2 or M phase, where there are inbuilt mechanisms that would check the cells for some abnormalities. If there are some abnormalities, then either if there are abnormalities and cells have ability to correct, then cells will go into G0 phase. And there could be a situation where cells cannot be repaired, then cells could undergo apoptosis, that is cell death. So cell cycle is very important to maintain the homeostasis of our cell. Right? So what did we learn here? Proto-oncogene, oncogenes, tumor suppressor, and checkpoints. Now let's look at few modifications that can occur on certain amino acid. What are these modifications? These are chemical modification on amino acid lysine, right? And these modifications are called as post-translational modification. Why they are called as post-translational modification? They're called as post-translational modifications because they occur after translation. So one of the modification is called as acetylation of amino acid lysine. Right? Then the second kind of mutation, modification could be methylation, right, of amino acid lysine. In context of cell cycle, phosphorylation is very important. Phosphorylation of serine, threonine, and tyrosine. These are the three amino acids which have OH group, right? And they have the ability to undergo phosphorylation. So what is that protein that triggers phosphorylation of serine, threonine, and tyrosine? Those proteins are called as kinases, right? So acetylation is triggered by lysine acetyl transferases. Methylation is triggered by methyl transferases, right? And phosphorylation is mediated by an enzyme called as kinases. And there is another modification which is very important for cell cycle. It's called as ubiquitination. This is mediated by enzyme called as ubiquitinase. Right? So these are the new terminologies that I discussed with you. I'll go over it in just a second. So these new terminologies are proto-oncogenes. These are the genes which stimulate growth. Oncogenes are the mutant product of proto-oncogenes, which will have the energy to drive uncontrolled cell division. Tumor suppressor are those proteins that would suppress the cell cycle to correct the mutation or problem and and put the cells back to the cell cycle. Checkpoints are those points in the transition phase of the cell cycle, G1, S, G2, and M, to correct any problem. Then these are post-translational modification. Acetylation occurs on amino acid lysine. Methylation occurs on amino acid lysine also, but they're different enzyme. Acetylation by lysine acetyltransferase, methylation by lysine methyltransferases. Phosphorylation occurs on three types of amino acid: serine, threonine, and tyrosine. And this is carried out by an enzyme called as kinases. And then the last one is ubiquitination. So for the point of cell cycle, we need to remember two kinds of modification: post-translational modification, phosphorylation, and ubiquitination. Now, cell cycle, as I mentioned to you, they move in phases, right? G1, S, G2, M. So these phases, going from G1 to S, G2 to M, and S to G2, they are controlled by a pair of proteins which are called as cyclin-dependent kinases and cyclin. So pay attention, this is very important. So hold on to the those terminologies: proto-oncogene, tumor suppressor, oncogene, acetylation, methylation, and phosphorylation and ubiquitination. And then let's look at some of the models of carcinogenesis. So this is a very very important and very powerful slide. So this is a normal colon wall. If there is a mutation in APC gene, anaphase promoting complex, then it leads to the formation of benign polyp, right? And it has been seen that if there is a mutation at one point, it can create a hotspot for another mutation. So with this APC mutation, there is a incorporation of RAS mutation. And what is RAS? RAS is an proto-oncogene. So if RAS is mutated, it becomes an oncogene, and which activity of RAS then again accelerates the growth of this benign polyp into adenoma. Further, there is a loss of DCC. What does DCC stands for? Deleted in colon cancer. So when this first, second, third mutation occurs, the growth becomes large and the benign tumor becomes adenoma. And then in the end, there is loss of tumor suppressor protein, P53, and that makes the tumor malignant and invasive. Okay. So what are the characteristics of cancer cells? They lose contact inhibition. So when cells, they come in contact with each other, they stop dividing, right? Because they don't want to divide over each other, right? That would create a problem. But cancer cells, they don't have this sense to stop dividing when they come in contact, right? Loss of saturation density. So there is a proportion in which cell division can continue to occur. Cell division has to stop when there is a certain amount of density. But these cancer cells loses the sense to see how much saturation is there, and they keep dividing. Independent of the presence of growth factor. What does this mean? Is that cells, they need growth factor to divide, but cancer cells, they have such a robust mutations that they don't require growth factor. They become independent of growth factor. They can become independent of oxygen because there are certain genes that can be activated and which leads to continued growth of cancer cells. Telomerase gene is activated, right? So if telomerase gene is activated, what will telomerase gene do? It will keep on adding, keep on elongating the ends of the chromosome, and the chromosomes become bigger and bigger, And if the chromosomes become bigger, then the anaphase, telophase, those phase of the mitosis or meiosis will be problematic. Right? APC, anaphase promoting complex, it promotes exit of mitosis from mitotic cycle, but if there is a problem, then the cell cycle will have abnormality. Okay? So let's go to the this slide, which again reinforces my re-emphasizes my point from earlier slide, that there can be a single mutation. This mutation can affect the neighboring cells, leading to the accumulation of second mutations, right? And then it becomes a hot spot for more mutations. So what happens? Cell survival and proliferation, a second mutation will further increase the give advantage to cell growth, cell invasiveness, and ultimately there will be metastasis of these tumor cells. Okay. Now I want to go come back to the to those two proteins which are called as cyclin-dependent kinases and cyclins, right? So these two are different proteins, cyclin-dependent kinases and cyclins, right? So cyclin-dependent kinases, what is the word here? Kinases. So these are enzymes, right? These kinases do what? They phosphorylate. Which amino acid could be phosphorylated? Serine, threonine, and tyrosine. And what are the cyclins? Cyclins are proteins which whose concentration changes with the stage of cell cycle, right? So CDKs, the level of CDKs in the cell cycle, it remains constant, but the level of cyclin is stage dependent, right? And because of this, these two proteins, they come together for the successful completion of cell cycle. So let's go to the next slide. So let's focus on the first chart. So during the mitosis phase, you see here, cyclin concentration increases. When the mitosis phases occur and the cells goes into interphase, the cyclin concentration dips, and then again cyclin concentration increases, and it is the highest in the mitosis and the lowest in the interphase. What are the interphase? G1, S phase, and G2 phase. But having less cyclin during interphase doesn't mean that there are low cycles. So let's look at this table. This table has G1 phase, S phase, G2 phase, and M phase. So a cyclin G1 will be from G1 phase to M phase. The G1S cyclin will only be at the boundary on G1 and S phase. S phase cyclin will be highest in the transition of S and G2 phase, and then the M cyclin. So these cyclins are the proteins which vary during each phase or during the transition of one phase to the other. I repeat that these cyclins, the concentration, the amount of these cyclins, they vary during each phase, particularly during the transition phase. If you note, G1 to S, S to G2, and G2 to M, right? And this change in concentration helps the cells to progress in the cell cycle, right? But what happens that cyclin, so let's look here more carefully. During G1 and S phase, right, there is cyclin E. During S to G2 phase, there is cyclin A. And during G2 to M phase, cyclin B. And M to G1 phase, cyclin D. So each different phases have different cyclin. And these cyclins, they help to make progress of the cell cycle, right? So these cyclins, they work together with CDK, right? So what are CDK? Cyclin-dependent kinases, right? So for example, here, cyclin E can partner with CDK2. Cyclin A can partner with CDK2. Cyclin B can partner with CDK1, right? So, so cyclin and cyclin-dependent kinases, they work together for the progress of cell cycle. Now, if the cells are not dividing, in a non-dividing cells, or the cell says pause, we don't need cyclin, right? The cell has to repair itself, or the cells are in G1 phase, or G0 phase, not G1 phase. So cyclin is not needed. So how cyclin is removed from the system? Cyclin is removed by a modification that I discussed earlier called as ubiquitination or ubiquitination. Ubiquitination of lysine, it flags the cyclins to undergo degradation, right? So you have to look at the whole story, right? So let's say if there is a problem in the G1S phase, the cells cannot go into S phase or G2 phase. If it goes, then there are chances that the cells can become problematic. So the cell cycle has to stop for the cells to enter into G0 phase for repair. So we don't need cyclin at that point. So this cyclin has to be removed. How this has to be removed? Because if it stays into the system, it will push the cell towards division. So it has to be removed. So it is removed through a biochemical process called as ubiquitination or ubiquitination. When this modification occurs on the lysines of cyclin, amino acid lysine of the cyclins, they flag this protein to undergo destruction. They undergo destruction by a mechanism called as proteasomal degradation, okay? And then the CDK, if the cyclin is not there, CDK becomes inactive, right? Cyclin has been degraded, CDK is inactive, there is no progression will be made towards cell cycle. Now, most of the cancerous drug, right? Most of the cancerous drug, or let's say, in other words, CDK is one of the target for cancer treatment. So CDK, cyclin dependent kinases, they phosphorylate the target protein. How do they phosphorylate? They phosphorylate by using the ATP as a source of energy. So what scientists are doing, they're trying to develop a compound that would mimic ATP, right? So if ATP is an important cofactor for CDK, and CDK has ability to trigger cancer progression, right? So if there is a drug company, a biotech company or pharmaceutical company that can develop analog of ATP, which no longer provide phosphate. What is ATP made up of? Adenosine triphosphate. ATP works as a cofactor and it donates its phosphorus for phosphorylation and it becomes ADP. So there's an analog of ATP, right? This can work as a medicine because it will inactivate the CDK. And if the CDK is inactivated, the cancer growth will be stopped. So one of the drugs is called as Gleevec, which is very effective in diseases such as leukemia. So how do Gleevec work? It mimics, or it's an ATP analog, and it goes and binds to the kinases. And when this Gleevec binds to the kinases, it cannot phosphorylate because it's not a ATP, it's not a cofactor. So cell cycle will stop. Right? So now here, there are three situations in which the cancer can occur. For example, by translocation, if a gene moves from one location to the new location, right? So if a gene is located adjacent to a promoter, and this is the natural promoter that controls the expression of the gene, but somehow this gene goes from the original promoter to the different promoter. The promoter has ability to express this gene differently. So this is going to affect the growth pattern. The second way a cancer can occur, then there can be gene amplification. So one gene is enough to carry out the function, but due to some abnormalities, instead of one copy of gene, there are multiple copy of gene, and there are several gene products. Then it will again push the cells towards dividing more. Point mutations. A single base mutation can convert a proto-oncogene into an oncogene, and then oncogene will stimulate the growth. Similarly, a point mutation in tumor suppressor also. A tumor suppressor has a job to control the growth, to repress the growth in case of problem. But if there is a mutation and tumor suppressor is not working as a suppressor, it becomes a growth stimulator, same like an oncogene. So mutations in both are very, very unrequired and dangerous. So there is a balance between oncogene product and tumor suppressor gene product to stimulate or to control cell cycle. Let's look at some of the questions in the end. So question number 1, if mammalian cells receive a go-ahead signal at G1 checkpoint, they will move into, so they get a receive go-ahead signal in G1 phase, so they will go into S phase and then G2 phase. So they will complete the cell cycle and divide, right? Question number 2, cells that are in a non-dividing state, what are that phase called as? So a non-dividing phase is called as G0 phase. Let's look at another question. What causes decrease in the amount of cyclin at a specific point in the cell cycle? So this decrease in cell cyclin amount will occur because of ubiquitination. Ubiquitination is a process where an ubiquitin chain is attached to the cyclins. And when ubiquitins are attached to the cyclins get flagged off and they are removed by proteasomal degradation machinery. So let's focus on this question. Study the following figure. In the figure above, mitosis is represented by which number? So let's say if 1 is G1 phase, this 2 is G2 phase. I repeat, 1 is G1 phase, 2 is S phase, 3 is G2 phase, 4th is mitosis phase. So mitosis is represented by D, 4th. Let's look at the next question. G1 is represented by which phase? So if this is G1S, G2M, G1. So G1 is represented by 1 and 5th phases in this diagram. Which number is represented by, which number represents DNA synthesis? So G1S, G2M. So S 2 it becomes S phase. Right? Question number, this question, which number represents the point in the cell cycle during which the chromosomes are replicated? So chromosomes are replicated in S phase. So G1, S, G2, M, G1. So the second phase represents the S phase where the chromosomes are replicated. Last question, which of the following are true concerning the cancer cells? They do not exhibit density dependent inhibition. True. When they stop dividing, they do at random points in the cell cycle. That's also true. They're not subject to cell cycle control. So all A, B, C are correct. The last question, which of the following are true concerning CDKs? CDK is inactive or turned off in the presence of cell cycle. It is wrong. CDK is active in presence of cell cycle. CDK is present throughout the cell cycle. That is right. It is cyclin that goes up and down. CDK, they represent constant. CDK is an enzyme that attaches phosphate group to the other proteins. That's right. CDK is a kinase. A kinase is an enzyme that can phosphorylate a target protein. And what cofactor they use? They use ATP. So B and C are true. A is false. Why it is false? Because CDK is always active in presence of cyclin. So with this, I finish this chapter on cell cycle carcinogenesis.